I think you just heard that message now that it's being recorded. And then I think that's it. Oh yeah, looks like pretty much everyone's doing this right now anyway, but keeping their video off during the presentations is, I think, preferable. And then when we get into the discussion period, I think if people want to turn their video on, certainly their microphones, that'd be the time to do that. Okay. So we're mostly on time here. I want to move on now to our first presentation here with Kate Reinl is going to talk about the Lake Superior Coordinated Science and Monitoring Initiative, just an introduction to what CSMI is and an overview of the CSMI focal areas. So Kate, it looks like you're sharing your screen. I'll turn it over to you.
Awesome, thanks, Matt. So yeah, I think I've met a lot of you formally or informally, but I'm Kate Reinl. I'm a PhD student over at UMD at the Large Lakes Observatory with Bob Sterner. I've been doing a lot of work on the algal blooms. I know starting to do some help with the CSMI planning. So myself, Anna Baker over at USGS, and Tom Hohenhorst have kind of been leading the parade on, you know, putting together all these efforts for CSMI, specifically for the cyanobacteria work. So this is going to be a pretty short presentation because a lot of you have probably already seen this material. So I'm going to keep it short and sweet. So if you need more elaboration, feel free to reach out to myself, Anna, or Tom. Let's see. I might have to click. Okay, there we go. So just a really brief overview of what CSMI is. So it's the Cooperative Science and Monitoring Initiative. It's the part of the science annex of the Great Lakes Restoration Initiative. And it works on a rotational basis. So it focuses on one of each of the Great Lakes each year. So there's 5 different stages, and those are pictured here at the bottom. So there's a field year, which is where Lake Superior is now, data analysis, report out, priority setting, and planning. So in 2019, we had this big meeting hosted by Minnesota Sea Grant to determine, what were the things that we needed to focus on for Lake Superior for the field year. And a lot of what came out was cyanobacteria work. So that's what you're going to be hearing a lot about today. So these priorities are set under the LAMPs. So if you're not familiar with the terminology, that's the lake-wide action management plans for each lake. And again, this is the intensive sampling year for Lake Superior. So the projects that I'll be talking about are going to be CSMI, but like Matt said, there's a lot of other work that's going on that's not funded by CSMI that's really kind of focused on the same things. Oh, I've got a chat. There's a lot of things. Okay, here we go. Let's see here. In December last year, we held this big meeting to really just kind of get the ball rolling with all of this. So this is a picture of many of your lovely faces that participated in that meeting. It was really great. We had lots of people from agencies, universities, that were participating in that meeting, giving overviews and really kind of just starting this whole process of building this collaboration in CSMI, where we aren't just siloing out all of these projects, and we're really kind of trying to integrate the work that we're doing so we can maximize our sampling efforts and also have this really nice cohesive data set at the end of all of these efforts. So I'm sure a lot of you are familiar, but in case you aren't, Lake Superior is not immune to cyanobacterial blooms, so we have seen them in varying size and duration since 2012, and there are some anecdotal historical observations, and those are shown with these open circles over here. But predominantly, they've been focused on the South Shore, as Matt said, on the Wisconsin shoreline. So the biggest blooms that we've observed so far have happened in 2012 and 2018, and those were years where we had those major flood events. And then we've also actually had a couple of reports in the last couple of years on the North Shore up here. So some of the previous work that's been done has shown that as I said, the biggest blooms happen in years with these rainfall events. And so we don't know exactly what that connection is. Bob Sterner's lab is currently working on that. His master's student, Tesha Coker, is doing some work looking at sediment contributions and nutrient contributions from storm events and how they contribute to blooms, but there seems to be some sort of connection there. And then we also saw that blooms occurred in relatively warmer years, and this is a figure over here from Bob's recent paper in LNO And we can put a link to that in the chat. I don't know, Bob, if you're on, if you want to throw that in there. It's an open access paper if you want to take a look at it. And then there's also been some experimental work done by myself in Northland that indicates that the species that we see in Lake Superior, Dilacospermum limmermanii, is or prefers low NDP conditions, so low nitrogen, high phosphorus conditions, and warmer temperatures. And that work also indicated that upland locations are likely sources or important locations for seeding blooms, and not necessarily that the lake is operating, completely, away from the landscape biologically and only receiving nutrients. It seems that there's a biological loading component as well. Let's see, what are we on from here? And I can also post that paper in the chat. So the CSMI funded projects, these are the cyano work ones that we'll mostly be hitting on today during the lightning talk. So these are under the land priority nutrients and bacterial pollution. So the kind of different subsections are tributary monitoring and phosphorus cycling, nearshore monitoring, mechanistic bloom drivers, and historical analysis. But we also wanted to bring up a few of these other projects that are going on in CSMI that are not necessarily under the cyanobacteria umbrella. So under the land priority habitat and species, we have these multi-trophic level assessment that's being performed by USGS and EPA, a groundwater study that's being done by USGS, food web dynamics, and coastal brook trout, which is being done by Michigan DNR, the Lake Sturgeon Index Survey, which is being done by the U.S. Fish and Wildlife Service, and then coastal wetlands study effects on water level also being done by the U.S. Fish and Wildlife Service. And then we also have some chemical contaminants work that's being done, which is going to be including some mercury monitoring. And so this is not an exhaustive list. This is just some of the work that's going on for CSMI specifically, or CSMI funded projects specifically. So we had that December 4th meeting, and we talked about those, different headings I just showed in the previous slide. And we talked about all the different projects going on, and we kind of developed these different subgroups around those different projects. And I'm not going to get into much to what these projects or what these subgroups are focusing on, because they're going to be getting lightning talks in just a moment. But the different subgroups that came out were nearshore monitoring, which is being led by Ellen Cooney over at Wisconsin DNR, tributary monitoring, which is being led by Anna Baker at USGS, microbial genetics in the open lake, which is being led by Abir over at EPA, remote sensing, which is being led by Tom also over at EPA, and then also a data integration and synthesis piece, which is being led by myself in partnership with the EPA. And so you're, over the next hour, I guess, we're going to hear from all of these different subgroups and the different projects that they have going on for CSMI. Just something else to mention, and I'll go over this sort of the data integration piece, but something else that has come out of this is that Tom and I over at EPA, Tom Holenhurst at EPA, and I have developed this ArcGIS online site where we've developed these interactive maps. And so this is just a screenshot. I'll show a little bit more during my lightning talk, but it's showing all the different CSMI planning that's been going on. And also we've been using this map as a tool to make adjustments so we can build a more integrated data set and really fine tune the sampling piece. So this is just kind of a sneak peek at some of what you're going to be seeing coming up. And I think that's all I have for here. So I will go ahead and stop sharing my screen. Thanks, everybody.
Thanks, Kate. That was great. All right, so let's move into the lightning talks then for each of the CSMI subgroup leads working on the cyanobacteria bloom issue. So first we have Ellen Cooney with the Wisconsin Department of Natural Resources, who will be talking to us about the nearshore monitoring subgroup. So Ellen, are you, oh, okay, great. We can see your screen.
You can see the presentation?
Yep, I can see it. So the mic is over to you.
Awesome. Okay. Good morning, everybody. I am Ellen Cooney with the Wisconsin DNR, and this morning I'm going to be chatting with you guys about the Nearshore Water Quality Group that we've established as a subgroup as part of the CSMI efforts. And so this group consists of anybody who will be monitoring water quality and algal parameters in situ sampling of the water column in the nearshore. And so this includes both the CSMI and non-CSMI projects. And some of the goals of this group were to coordinate it between the partners on sampling parameters, timing, location, as well as methods, with the overall objective of creating a sampling designed to inform us of these nearshore dynamics and how they're going to be how they could potentially influence algal blooms. There are many different projects that are occurring in the nearshore this summer, and in the following slides, I'm just going to give a quick snapshot of some of that work. All right. So one of the projects that is a lot of coordination has occurred between the Large Lakes Observatory at UMD and the Burke Center at Northland College, as well as Wisconsin DNR. We're working to characterize the chemical, physical, and biological characteristics of Wisconsin's Lake Superior nearshore waters, both spatially and temporally. We're hoping to identify key characteristics or signals in the nearshore to support efforts in identifying the mechanistic drivers of these blooms. And so the Large Lakes Observatory at UMD and the Burke Center are collaborating to collect the zone profiles as well as wet chemistry surface grabs for chlorophyll, particulate and dissolved nutrients, suspended sediments, silica, microbial DNA sequencing at approximately 6 time points between mid-June and mid-September. And the sampling approach is designed to give a picture of those nearshore conditions from the tributary mouths to offshore. And the Wisconsin DNR effort is looking at a more longitudinal study, sampling across the from about Superior to Muwekwe Bay. The UMD Northland College sampling is also going to include areas in the Chequamegon Bay in an effort to help understand those spatial differences in water chemistry and microbial community to understand the bloom risks there. And so that project ties into the Wisconsin DNR sampling, which will be done in a similar time frame from about mid-June to mid-September, and we'll be sampling about every 2 weeks. Similar parameters for sampling, except for the silica. And then in a bloom response, we'll have some phycocyan and saxitoxin and anotoxin, as well as algal ID. And from those same 15 sampling points that Wisconsin DNR will be collecting, that water will be used in a nutrient uptake analysis that will be performed by the EPA GL-TED lab. And Terry will be using those samples about once a month, one pre-bloom, one during bloom, and then one in August and September timeframe as well. And this is the map that Kate just showed in her previous slide here. So we have, this is kind of a spatial distribution of all those sampling points that'll be going on in the nearshore. And these dotted lines here are the in situ kind of event-driven sampling that will be occurring by USGS. They hope to conduct about 2 event-driven water quality and hydrodynamic surveys from Superior to Wisconsin, or Superior, Wisconsin, to Myers Beach. And they'll be emphasizing capturing runoff events at the Siskiwit River and Saxon Creek. This will integrate with the continuous tow data, as well as the Wisconsin DNR water quality sampling efforts. There are 2 different towing options, and we may be able to hear more later as to which one we might be leaning towards. But there are a couple of different survey designs here on the map to see. The other projects that are going on in the area are the National Park Service. And they will be doing some water quality within 3 nearshore sites within and upstream of the Apostle Islands, looking at water quality sampling, as well as continuous sensor deployment in Siskiwit and Muwekwe Bay. And the final project that we're working on is the Northland College. They're working on to determine the potential source for cyanobacteria blooms in Chequamegon Bay. So we'll have some more time later for discussions, but those are some of the main projects that we're coordinating this summer. So I'm all done, and who is next?
Great. Thanks a bunch, Ellen. A lot of information there in a short amount of time, so appreciate that.
Speed reading.
And we do have some time for questions for all the CSMI subgroup leads here at once we get through all the lightning talks. So save your questions. So next we have Anna Baker from the U.S. Geological Survey is going to talk about the tributary monitoring subgroup. So Anna, whenever you're ready, if you can share your screen. All right, we can see your screen.
You're seeing it? Okay, great.
Yep.
Morning, everybody. I'm Anna Baker. I'm a hydrologist with USGS, and I've been working with Eric Danswine to co-lead the tributary monitoring and modeling subgroup. This group involves a large group of researchers. The representatives you see listed here are coordinating on this subgroup on behalf of an even larger group of folks, and all coming from the agencies you see listed at the bottom here. So we wanted to give just a really high-level overview of some of the research questions that are driving the projects that have been developed and are being coordinated through the tributary monitoring and modeling subgroup. So we'll start out with some of our research questions. We're interested in how the timing and magnitude of loads of nutrients, sediment, and cyanobacteria from tributaries may be influencing nearshore cyanobacterial blooms. We're also interested in where hotspots for nutrient, sediment, and cyanobacterial introduction may occur throughout the watershed. We'll be looking at the role that sediments play in in-stream nutrient cycling, and we're also curious about how sediments from the watershed may be contributing to legacy phosphorus development in the nearshore. We'll be looking at how microbial nutrient uptake and spiraling in tributaries may affect downstream nutrient levels in cyanobacterial populations, and also a little bit at the stoichiometric requirements for driving cyanobacterial growth in these settings. And lastly, we're interested in where in the watershed targeted management might have the greatest potential to reduce nutrient and sediment loads coming out of the watershed and subsequently reduce the occurrence of nearshore cyanobacterial blooms. So with those questions in mind, we have a bunch of projects taking place that are led by this large group of researchers. And they're taking place kind of across the South Shore western arm of Lake Superior, but there's a really strong focus on the Siskiwit River Basin. And the Siskiwit has been shown in previous studies to be a potential source of nutrients and cyanobacteria to the near shore, and there have been repeated bloom occurrences in Siskiwit Bay and also Myers Beach and the Apostle Islands to the east. And so the potential for transport of nutrient, sediment, and cyanobacteria from this river system and watershed through the near shore is strongly of interest. So a few of our projects taking place here include the monitoring of algal composition and algal toxins, as well as nutrient concentrations and temperature from Siskiwit Lake and also downstream near the river mouth. And that is a collaboration between folks at UMD and National Park Service. We also have a group from USGS that will be monitoring storm flow and base flow nutrient and sediment loads in the upstream portion of the Siskiwit and also downstream. A portion of this project will also be devoted to looking at the nutrient exchange potential of suspended sediment and stream bed sediments. And we're also hoping to have a component of the study that focuses on the microbial community in sediment and water under these varied hydrologic conditions. An EPA-led study is going to conduct in-stream nutrient uptake assays to look at microbial exchange of nutrients. And as part of that project, EPA will also be characterizing the microbial community using genetic signatures under these varying controlled nutrient conditions. And lastly, a collaborative project between EPA and Natural Resources Research Institute will be constructing a watershed model using the VELMA framework. And VELMA is an EPA modeling framework. VELMA is an acronym for Visualizing Ecosystem Land Management Assessments. And this model predicts the effectiveness of alternative land use and land cover scenarios for protecting water quality, and also estimates potential ecosystem co-benefits and trade-offs. And this model was really designed mostly to look at in-stream nitrogen cycling, but this group will be developing modules for both sediment and phosphorus. There also are a number of related ongoing studies that have been coordinating with the CSMI studies through the Tributary Monitoring and Modeling subgroup, and we wanted to highlight some of those here. The Red Cliff Band Water Resources Program, which is led by Ali Rakowski, is working on developing a water quality baseline in 7 tributaries and the Nearshore. So these are tributaries that drain through Red Cliff tribal lands in the Bayfield Peninsula. Also, as previously mentioned, graduate student Tesha Coker from University of Minnesota Duluth is looking at the connection between storm transported sediment and cyanobacterial blooms. So sampling sediment at a network of tributaries spanning from the Nemadji over to the Bayfield Peninsula and looking at the phosphorus content and how that may be connected to cyanobacterial blooms. And Rob Mooney is also coordinating with this subgroup, and he's going to share some of the details of his work with the Northeast Climate Adaptation Center in a separate lightning talk, but they'll be doing sediment cores, looking at the history of cyanobacterial blooms as well as water quality sampling. So we've been working to try and coordinate across all of these great projects taking place this season, and we look forward to sharing more nitty-gritty details and discussing some of that planning in the discussion section. So thanks.
Great, thanks a lot, Anna. And just so everyone knows, Val put in the chat, if you do have questions that you want to address during the question and answer session, you can either save them for the end or type them into the chat and we can get to them that way as well. So next we have Abir Banerjee from the US EPA, who's going to talk with us about the microbe subgroup. And Abir, you don't have slides, correct? You're just going to talk?
Correct. Thanks, Matt. Yeah, folks, I apologize. I was too lazy to make slides. And actually, after hearing from Ellen and Anna, I'm kind of glad that I didn't make them. Originally, I was also going to pull up the same interactive map so that everybody could visualize the sampling areas, and I'd go over what our group is doing in terms of locations and timings. But again, I think Ellen and Anna have already done a fantastic job of covering those bases. So what I'm going to do instead is take this opportunity to give a shout out to my group and to go into a little bit more details about our methodologies and what kinds of questions we're hoping to address and how we're supporting the other initiatives. Later on, you'll get a chance to hear some of the additional collaborations that some of our group members are coordinating with Bob Sterner's group. So I'm not going to steal their thunder on that topic either. Backing up, my microbe subgroup comprises myself. I'm Abir Banerjee from the US EPA, Matt Hudson, who you all know as well. We've got Carrie Givens from the USGS and Richard Kiesling, also USGS, Kyoko Yokota from SUNY, and Ewan Reevy and Cody Sheik from University of Minnesota Duluth. As Anna had already mentioned, our group's main focus is trying to characterize the microbial communities associated with these bloom events and the conditions surrounding them. We have multiple avenues where we are coordinating with the other data collection so that we can, by the time all said and done, be able to address the questions of how these blooms form, what's happening in terms of the cyanobacterial dispersal, and also perhaps get some inferences about the ecological interactions that the cyanobacteria have with other microbes and with the environmental conditions surrounding their growth. A lot of our methods are going to be heavily genetic based. We're going to be doing DNA extractions and implementing things like metabarcoding or eDNA. And the goal in that is to get a good sort of handle on the community profile of these microbes, which aren't necessarily culturable by standard methods and aren't usually on the radar screen if we're trying to monitor blooms. The logic behind that is multiple fold. We've got the possibility of having a better understanding of the actual population dynamics of the cyanobacteria, what kind of demography there is. We might be able to distinguish between toxin producers and not if it comes up. And we might be able to have early detection aspects of it, depending on the sequence of events that happen with the sampling. You know, there are microbial signatures associated with certain kinds of inputs like agricultural runoff versus urban settings and that kind of stuff. Our group will hopefully be able to put those pieces together to tell a nice story about how these things work and get at a better mechanistic understanding of what goes on with these blooms. That's all I got. Thanks.
Great, thanks, Abir. Appreciate that. All right, next we're going to move to Tom Hollenhorst with the US EPA. He's going to talk about the remote sensing subgroup. All right, Tom, over to you. I can see your screen.
All right, can you hear me?
And I can hear you.
See if I can get this to go into presentation mode. Hello, everyone. It's really exciting to hear about all this work coming together. I think it's really exciting. The potential we have for the coming field season and integrating all of this data for a better understanding of how these blooms form. So I've been heading up the remote sensing group. We have these objectives of employing remotely sensed data to identify past and current algae bloom occurrences. We'd also like to identify and characterize the tributary blooms and the sediment movements along the shore using remotely sensed imagery. We're working to synchronize in situ sampling with satellite orbits and schedules. And I'll talk a little bit more about that. And then we're working with some EPA ORD scientists, Daryl Keith and others, to develop and calibrate algorithms and techniques for classifying HABs using the in situ data from the gliders we'll be deploying, the towed arrays that Paul Renault will be operating, and all of the nearshore sampling. We're also looking to provide near real-time information for all of the different field researchers for information about the tributary plumes and the blooms, and to employ that remotely sensed data for adaptive in situ sampling when we see a bloom occur, so that we can sample inside that bloom and outside and around the edges. Also looking at new sources for imagery, and also expanding on the CYAN app for early detection of HABs and cyanobacteria blooms. This is a satellite scene from the Sentinel-3 satellite. It's from March 19th. And you can already see that there's quite a bit of sediment moving around in the near shore of the lake. And sometimes it seems pretty direct. You can see which tributaries it might be associated with. You can see a plume here that might be associated with the Nemadji River, but maybe some resuspension along the south shore as well. How we quantify that and characterize that is going to be the trick. And I'm looking forward to some of the expertise that Daryl Keith and others are bringing to the table in that regard. I really, really wish Michelle was here to be with us and see all of this great collaboration occurring. But back in 2017, she sent a table similar to this table here on the left, showing the path and the dates that Landsat 7, Landsat 8 would be above as people were sampling in the nearshore. I went back to emails from her from back then and was able to recreate what she had done. And now we have this predictive schedule of when Landsat 7, 8, Sentinel-2, A and B will be overhead. Landsat 7 revisits every 16 days, as does Landsat 8. The Sentinel-2 data revisits every 10 days, but there's 2 up there, so we'll have Sentinel-2 overhead flights every 5 days. So we have this schedule laid out from middle of March to the end of September. But if people are out sampling past that point, we can extend that as well. And right now, this schedule is for for Landsat, this row 26 path, or path 26, row 27, which just misses this Duluth area here. But these other satellites right in conjunction will be very close in time. The Sentinel-3 data occurs nearly daily, as does the MODIS data. So we'll always have that available too at a coarser resolution. We'll be making this available on the hub site that Kate mentioned earlier. So then just quickly talking about the CYAN app. This is what Daryl Keith and others at EPA ORD have been developing with collaboration with folks at NASA and elsewhere. They have developed an algorithm for predicting cyanobacteria and microcystin cell counts from MODIS and Sentinel-3 data. And it's available across the lower 48 of the United States. So for any lake larger than a certain size, I think it's about 5 or 10 acres, you can put a pin on that lake and see what the predicted cell count, microcystin cell count is for that time. It also makes available a time series showing what that cell count might have been in the past with some, I'm not sure it gives you a predictive, but we can at least start to see how these blooms are developing. So far, this app because of how the satellite scenes were acquired and applied, is available in some parts of the nearshore in the Great Lakes, but not the majority of the Great Lakes. So Daryl and others will be working to plug in that hole and make the CYAN app applicable in Lake Superior, especially along the South Shore. On the bottom left, I think my slide got cut off a little bit, but it's the chlorophyll from and predicting cell count, and then Secchi depth related to chlorophyll as well. And that's something we're interested in learning about. Maybe we can talk about during the discussion period too, is is there anybody that will be collecting Secchi depth information? Because that would be useful for Daryl and his team as they're developing these algorithms for predicting the cyanobacteria microcystin cell counts. That's all I have. Thanks.
Great, thanks, Tom. All right, so now to the final lightning talk for CSMI subgroups. It's back to Kate Reinl, who will talk about the data integration piece associated. This is a big effort. So Kate. And I can see your slides.
Totally forgot to unmute myself there. Thanks, Matt.
Yeah.
So, we don't have a lot of, projects similar to the other subgroups because we're working with kind of bringing all that together. But there are three kind of main components that have fallen underneath of my subgroup's umbrella. So the first is assisting in developing collaborative sampling plans among agencies and institutions, and that's what you're seeing right now. All of this work that's being done by myself, Tom, and Anna. We've been, getting these meetings together, developing these subgroups, and as we've been calling it, herding cats, and kind of getting everybody to work together to develop these collaborative plans. But part of that has also been developing this tool, this ArcGIS online site, the hub site. And I'm really, really, really happy to see that so many people have been using it in their planning. I'm really, really glad that it's been a great tool to use. And so that was the main purpose of it, was just developing that map, but then, it expanded into the site that we're using for planning as well as public engagement. Another component to this subgroup is developing plans for the interlab calibration. I should have put the formalized name on here, the Lake Superior Interlab Calibration Effort, or Lake Superior ICE, which is being led by Joel Hoffman and Chris Philstrip as well. The sensor component is being led by Kansas Keaton over at EPA. Joel and Chris aren't really able to be in on portions of this call, so I'm kind of going to try to retrieve the information they're looking for, but they're hoping to kind of get this effort off of the ground, and we'll talk about that during the discussion period, and I can discuss more of that later. But basically working to do sort of like a round robin style sample swapping so that we can make sure that our data actually is compatible through our lab analyses. So we want to make sure that our wet chemistry matches up. So we want to swap samples for, nutrients and pigments especially, as well as making sure that our sensor data is calibrated, and cross-checking to make sure that that information lines up as well. And then the last component of this is piloting a data integration system for CSMI using highly detailed metadata. So one of the big problems with CSMI data and data in general is that it gets collected and then it gets buried, and a lot of people aren't able to find it. So I'm working on putting together a platform. Right now we're looking at using GitHub to use a highly detailed metadata for looking at what data is available and how to retrieve it. And those efforts are going to really kind of get going more as the summer moves along. Let me see here. Okay, so I just pulled up all the different, can you see the new screen, Matt, or somebody, could you chime in?
Yep.
Yes.
Yes. Okay, great. So this is the hub site that we've been talking about. I just pulled up all the tabs up here just so in case my internet connection isn't stable. So this is the CSMI 2021 Lake Superior Intensive Field Sampling Year. So this is the home page. It talks about what CSMI is, information on the planning workshops. We also have a tab for maps and data. I'm going to circle back to that at the end though. We have a location for publications. So publications that are related to CSMI where you can also submit publications. I don't really want to wait for all this stuff to load because I don't want to take up too much time, but you can actually submit publications that we don't have on here as well. We have a location for photos so that people can see what it looks like to be doing photo information or sorry, so people can see what we're doing during CSMI, kind of, you know, a cool public engagement thing. So we have these photos from past CSMI on here. We also are going to be posting citizen science and outreach information on here in cooperation with Minnesota and Wisconsin Sea Grant. And then we also have a contact us page. Kind of the crown jewel of this site though is the maps and data page. So this is the interactive map that we've been showing. This is kind of a different format for it that I'm using on the hub site right now. So you can actually click on these points and you can see the location, you can see when the sampling is happening, how often, you can see what lab parameters are being collected, the sensors, et cetera, different information like that. You can zoom in and out. You can also add your own data to this map to see how it lines up. And this is just one of the maps that I have located on here. We also have existing data, weather and climate, remote sensing. So this is just an example. We have, I have chlorophyll remote sensing data on here as well. So I'm really hoping for this to be a really great tool for everybody to use. I'd really like to hear if there's something that you think that it really needs that it, you know, to be useful to you. I'd love to hear from you guys about that. And I think that's all the time I really have. So I'll go ahead and stop sharing and thank you, everybody.
Great, thanks. Thanks a lot, Kate. You know, it might not be the worst thing to leave that interactive map up there, if you don't mind, while we get into the question period here.
Yeah, I can do that.
Okay. So it looks like Aaron shared the link for that ArcGIS online map in the chat. If folks are interested in following what's going on with the CSMI work, check that out. So now we have a few minutes for questions here related to the CSMI subgroup leads, and start my video here. So it looks like there was a few comments in the chat regarding the Siskiwit Basin project from Al Kirschbaum. Definitely great to hear the shout out to Michelle. She would be really excited to see all this work going on. And we definitely all miss not having her with us this year. I do have a couple specific questions, but I want to leave it open to others who might have questions for the CSMI subgroup leads. Okay. There was a question about, is anyone doing research experiments on toxin producing conditions? So Val mentioned there is one from Kansas Keaton's. Can we have someone address that question?
This is Kate Reinl. Right now, I don't think that we have anybody that's specifically looking at that. We do collect samples, at least through NPS and UMD sample collection, looking at toxins, just detection, not specific work targeted at understanding conditions related to toxins. I could be wrong. There might be some new projects going on. So if anybody does have that work going on, feel free to jump in.
I was about to say something along the same lines.
I think that's right.
Yeah.
Abir, did you have something you wanted to add there?
Yeah, no, I was just going to second what Kate said. No one's doing it specifically. Our group is interested in the mechanisms underlying it, but we're hoping to get at that indirectly by sort of comparing our data sets to the ones where we have data for the toxins if they show up. And I saw someone in the chat pointed out that we haven't had a good history of the toxins in our case, not in Superior.
Okay. And I know
I'll
Gina,
just add.
oh, go ahead, Brenda.
Yeah, sorry, this is Brenda. I was just going to add that in 2021, Apostle Islands is participating in a nationwide National Park Service USGS HABs project. And in addition to like the discrete toxin sampling, which we've done, as Kate mentioned in the past, we're also going to be deploying these spat samplers, which should integrate toxin, they'll absorb toxins over a period of time. And so those will also be analyzed this year, just from one site, from our Mowickwe Bay Myers Beach site. And, because they'll integrate over a broader amount of time, maybe we'll capture more than we have in the past. And because we're doing repeated sampling at those sites, we'll also have an opportunity to, match up any toxin hits with environmental data. So there's a little bit of potential there, more this coming year than in the past, to pick up on what toxin producing conditions might look like.
Great, thanks, Brenda. I have a note here that Tim Asplund had his hand up. Tim, do you have a question?
Yeah, thank you. This is Tim Asplund. I'm the water quality monitoring section chief in Madison for Wisconsin DNR. So thanks for kind of allowing me to listen in here. I'm not as familiar with all the details, so it's really great to see everything going on this next year. But I do have a question about the Siskiwit River Tributary Monitoring Project that's kind of highlighted right there. I think someone said, the presenter, that there was some evidence this might be a watershed that's contributing maybe higher nutrient levels and maybe a driver of some of the recent nearshore blooms. Can someone just go expand on that a little bit?
Sorry, I was looking for my unmute button. This is Anna. Previous work that's been done by Kate and Bob Sterner and others has looked at cyanobacteria propagules in this system. To my knowledge, there hasn't been a huge amount of studies of nutrient and sediment output from this system yet. And so we're hoping to get a good amount of data from this system this season. Kate and or Bob, did you want to speak to previous work in the system at all in addition to that?
Yeah, Bob Sterner here, thanks. There's a little bit of Siskiwit data in our limnology and oceanography paper that's already been referenced and is linked in the chat. Also, I have a slide pertaining to this in my upcoming lightning talk, where we have data from last year for the Siskiwit. Directly to the question, we don't really have any evidence that I can think of that the Siskiwit itself is a, kind of a major driver in this nearshore, but so much of that is linked to hydrodynamics that it's difficult to be definitive about it. It's not a very big river, but it's right there where all the action is. So that's all I've got for you.
Something I'll add. So Siskiwit River was one of the rivers that I analyzed in my fluvial seeding paper. And no, it wasn't necessarily the smoking gun. There wasn't really very many locations where it was like, off the charts, crazy growth. So we took samples from the rivers and brought them back to the lab. So if there were cyanobacteria in the river, we put them under a broad range of conditions. So if there was cyanobacteria there, it probably would have grown. Actually, one of the rivers that we did see a lot of growth in is Lost Creek #2 right here, which is another one. But through monitoring efforts, we have seen cyanobacteria in Siskiwit Lake. And as several people have mentioned, we've seen cyanobacteria in the Siskiwit Bay region. But as Bob said, you can look at the data from my paper. Siskiwit River was not necessarily like, this crazy hotspot where it's just chugging out cells or anything like that.
Thanks, Kate and Bob and Anna. I did have one quick question for Ellen related to the DNR nearshore sampling. Were you all going to be collecting samples for the microbial DNA sequencing?
Yeah, I think we were waiting to hear back if the materials were able to be provided for, because we weren't able to analyze them, but if there was funding, we would be able to collect water, yes.
Okay. So we will be through Bob and I with our work collecting those samples with our nearshore work and working with Cody Sheik in his lab. So we'll at least get some of that nearshore tributary mouth microbial sequencing. Okay, well, we're at just past 10 o'clock here. Other questions that come up, please put them in the chat. We will have another question answer session here after the next group of lightning talks, as well as there's time in the discussion to address things that are coming up here as we go. But right now I want to give us a short break here just until 10:10. So we'll leave the Zoom open here, but it will return at 10:10 and we'll continue the lightning talks at that time. So we'll see everyone back here in 8 minutes.
Sounds good, Matt. Thanks.
Brenda, are you online at the moment? All right, yes, you are. I was just gonna suggest you do that. Thank you.
You betcha. Does it look okay? Can you see it?
Yes, you're not in presentation mode, but we can, now you are, yep.
Great.
And I caught that you betcha there. It sounded a little like Charlie Barron's. That's how I committed.
I heard he's coming to Big Top this summer.
Oh boy,
Mhm.
I have to catch that one. All right, we'll just wait one more minute here.
I have to say, since moving to Minnesota, I'm a little disappointed that not everybody says you betcha in every sentence or ends their sentences with a. Kind of assumed they would.
Everybody has to do their part of you.
I'll work on it, develop my own accent.
All right, well, we're at 10:10 here, so why don't we keep going with the agenda? All right, so next we have Brenda LaFrancois with the National Park Service, who's going to share with us some updates on their invasive mussel work and some other related project work. So Brenda, I'll hand it over to you.
Right, thanks, Matt. And I'm going to be joined by Jay Glace from the Park Service too in a minute here. And we're talking about the work of various partners, some of which are represented with the logos on the slides here. But two projects that we wanted to update people on are our ongoing invasive mussel work in the Apostles, as well as our ongoing benthic mapping work. So these are just some brief project updates related to those. So on the invasive mussel front, I just wanted to share some recent findings from Mark Hovey and Tobin LaFrancois, as well as from Annette Trebitz's group at EPA and Mike McCartney, formerly at University of Minnesota. And what you're seeing here are a couple of graphics showing what appear to be some pretty strong spatial gradients in zebra mussel detections and abundance from west to east in the park. The graph on top shows what's going on with adult zebra mussels, where there's more of them in these shoal sites in the western part of the park. And the same thing was found in some villager work by Annette's group in the graph or the map at the bottom there, with some higher densities in the west there. And more recently, Annette's group from EPA has found surface gradients in eDNA as well from St. Louis River Estuary over toward the Apostle. So it seems like we do get detections throughout the park, but there does seem to be a concentration in the western part of the park, which might indicate that there's some introduction happening via drift from known populations in Duluth Superior. And that's consistent with some of Mike McCartney's genetic findings, which is showing some genetic similarity between mussels in the Duluth area and mussels in the Apostles relative to other areas. We are seeing them on heavily used docks and mooring areas. We're also finding them on native mussels, so they're colonizing a variety of substrates. And our partners from Northland and from U of M have shown that on a couple of these sites, a wreck site, as well as a couple of dock locations. It does seem like we're able to exert some control locally by doing manual removal efforts. So we're going to sustain those in the future. In 2021, Apostle Islands is going to be participating in a multi-park invasive mussel project throughout Lake Superior. Fish and Wildlife Service is helping us with some of the passive sampling method development and sample processing. And so we'll be doing some methods comparisons, looking at detections via scuba-based work versus passive samplers. We'll also be comparing the types of detections we're getting on different kinds of sites, like docks versus wrecks and reefs. And we'll be targeting quite a few sites in the Apostles in 2021, including probably some new sites along Long Island and potentially in Chequamegon Bay. That's all I have on that project. So I'll switch to you, Jay, if you unmute.
Yep.
talk about the
Okay.
benthic mapping work.
Thanks, Brenda. I think I'm unmuted. Can't hear me, just let me know. I think a lot of you are familiar with some of the bathymetric mapping that the Park Service has been doing for several years. So in 2020, NOAA worked with us to take over this mapping effort, and they used our vessel, the Echo that's pictured here on the top left of that slide, to continue mapping on the west side of the Bayfield Peninsula. And you can see Brenda has in the slides here that they've got an additional 30 square miles mapped for us in that dark gray area. And we're hoping this coming summer to get some ground truthing done with video and sediment sampling, and use that to define habitat under the CMEX classification standard, which is the Coastal and Marine Ecological Classification Standard that's been developed by NOAA, and it's being more and more widely used throughout the Great Lakes. And let's see, on to the next slide, Brenda. If you can hear me. There you go. So along with the habitat delineation that we're hoping to do, we'll have these geoformation maps that are modeled with a couple of different models here. On the left, you can see some work that was done near the Oak Island Shoal in the Apostles using the benthic terrain modeler. And on the right, a different part of the Apostles using another geoformation modeler. And I'm not going to go through what the acronym stands for because I honestly can't remember. But these give us some definitions of the geoformations on the lake bottom. And then combining that with the bottom types, the substrate types that we hope to get from the video and the sediment grabs, we'll hopefully come up with a way to better identify habitat throughout the region that was shown on that, actually throughout all of the areas of the Apostles where we have mapping and we have video imagery, but to use that to come up with habitat maps to help us better identify these areas where we might want to look for invasive and native mussels to connect it back to that invasive mussel stuff that Brenda was just talking about. And we're actually hoping that there will be multiple uses for the information. There are a couple of fisheries examples that we hope to use this for, specifically some work with Cisco behavior that's going to be going on in the Apostles this coming summer. And there's a little bit there about some new data collection potentially in Chequamegon Bay this year, and that's related again to some concerns about invasive mussels and the native mussel beds that we've found there on the inside of the Long Island area. And I think that's all I've got, unless you have anything else to add to that, Brenda.
Nope, that'll do it.
Great, thanks, Brenda and Jay. It looks like you got at least one question in the chat. Next, we're going to move to Bob Sterner. Bob's going to give us some results from their work with the National Park Service along the South Shore. So Bob, I'll hand it over to you.
Thanks, Matt. Let's see, I'm working on getting my screen shared. Sorry, not seeing the one I want.
If you need help, I have your talk up as well.
I'll just do the whole screen. Can you see me or see the screen?
Yep.
Okay. So yeah, thanks. You've heard a bit already about things we're involved in. And if someone at some point can tell me how to share a file, I typed up a little cheat sheet about all the things that UNB is doing and can share that. So it's just a list of the various projects and also the actual references to the papers we published so far. So it's a Word doc that I'll share once I figure out how to do that. I thought what would be most useful today in the few minutes I've got to just show you a little bit of unpublished data, mostly from last year, but also some more comprehensive analysis of the Brule that we've been doing. And by way of getting into that, we've been doing work in this region since 2017. Brenda came to us and said, hey, we were looking for somebody to take over some work that UW Milwaukee was stopping. And in 2017, I thought, oh, hardly anybody's studying the South Shore of Lake Superior, and it's an important sediment source. So this would be an interesting side project. And it's pretty incredible and a little bit daunting to think about where we are today compared to where we were back then. Here's the first little snippet. We have deployed moorings in the nearshore in several different years, and have been variously successful in getting those moorings to function and get the data off of them. 2020 was deployment was delayed because of COVID, and then we had some issues with some of the equipment, but we do have a little bit of data. So we've got moorings from Siskiwit Bay, from Mawikwi Bay. These are temperature recordings at 2 meters, 5 meters, and 9 meters for both of these deployments. And you can kind of see an alternation between periods of time when we've got stratification and periods of time when we don't. Mostly, I think, having to do with how the water is sloshing around out there. Turbidity sensors are, I think, going to be super helpful in a variety of ways. They're picking up, some real variability. The two moorings, even though they're pretty close to each other, are doing pretty different things with respect to turbidity, which isn't a huge surprise when you just look at enough aerial imagery. That's very highly variable environment out there. And then the chlorophyll sensors, I wanted to show you these. These are the same data, just stretched out. And the main thing I want to point out here is the really strong diurnal variability at 3.5 and 4.5 meters depth. The main thing which determines what quote unquote chlorophyll these recorders see is the time of day. So it's a really seriously challenging technical problem to get chlorophyll data off of these sensors in this environment. So there may be questions about that, but it's something to keep in mind. Let me go into presentation mode to use my animations. So here's some discrete water sample data. I mentioned I would talk about the Siskiwit River a little bit. So these are all data from last year for the sampling sites that are highlighted up here. And then this area is zoomed in down here just so you can see them better. We've got chlorophyll, TSS, TP, and TDP, and we've got other parameters that we've measured, but I picked these out today to show you. These are the nearshore sites, what I would call nearshore, the Bark Bay site, the Mawikwi Bay site, and the Siskiwit Bay site. Chlorophyll, TSS, TP, TDP. And one of the main messages here is just sort of how unvariable seasonally this environment is, even though we know there's sediment coming and going, and in general in big lakes, the nearshore is way more dynamic than the offshore. Still, in the grand scheme of things, it's really difficult to sort of identify anything about season in these data that you see here, but also in other data that we've collected in other years. The rivers, we have the Brule and the Siskiwit River here, and the Brule sampling last year included one pretty significant flow event here in these yellowish bars. So you see in the flood, there's elevated TSS, even elevated chlorophyll, TP, TDP maybe a little bit. The Siskiwit River, we didn't have such a big flow event. But one interesting thing that I'm just seeing in these data, the Siskiwit River seems to be putting out somewhat more TDP than, in terms of concentration, not loading, than the Brule. So we're seeing some differences in the Siskiwit versus the Brule rivers. Maybe subtle differences, but I think some pretty clear differences. This is the multi-year analysis of the Brule flow. So we've been sampling the Brule at the gauging station for a handful of years now. And these are plots of the chemistry in the Brule River as a function of the flow measured at that USGS gauging site. And here's TDP in the Brule. So when there's floods, there's more dissolved phosphorus in the water. We have TSS data only from 2020, but high flood means more stuff being carried downstream. Nitrogen is doing interesting things because ammonium and nitrate aren't affected much by flow, but there's a lot of organic nitrogen. This is TDN. So the difference between this and these others is organic nitrogen. So floods mean a big flush of organic nitrogen out of the Brule. The TP data here is interesting and looks like some other measurements of other parameters where at sort of low to low-ish flow, there seems to be a pretty consistent flow concentration relationship. But you get into these really big events and they just do crazy things. So these big flow events for us are dramatically undersampled events, just a grab sample on an opportunistic basis here and there. But I guess what I'm seeing in our data is that these really extreme events are not predictable from the concentration versus flow data from lower flow events. Big events are different, doing different things, moving stuff off the landscape different than a simple extrapolation would predict.
Hey, Bob.
Yeah.
This is Matt. We should probably have you wrap it up here real quick if you can.
This is the last slide.
Okay.
Tesha's been mentioned. So these are phosphorus concentrations in different sediment locations around the lake in work that she's doing. And she's doing some other measurements as well to try to get at the bioavailability of phosphorus in these samples. So sorry if I went over, that's it.
Sorry, everyone. Just having a hard time finding my unmute. Looks like Rob, you're getting ready with your slides. So I'll hand it over to you next. Rob Mooney's talking about, well, take it away, Rob.
Awesome, thanks. Yeah, so my name is Rob. I'm a postdoc with Hillary Dugan at Center for Limnology at UW-Madison. I'm actually a postdoc on a different project, but I've worked with NECASC before on some Great Lakes tributary stuff. So it's hard not to get involved with this project. It's looking at climate, storms, and the drivers of cyanobacteria blooms in Lake Superior. So the PIs are Hillary Dugan at UW, Bob Sterner, Catherine Schreiner, and Byron Steinman, all at the Large Lakes Observatory. And the two main objectives of the project that I'm going to be talking about, and so this is all going to be happening over the next year and a half, 2 years or so. And so it's really going to be mostly methods that I'm going to be talking about, but the main objectives are to determine if climate is driving these cyanobacterial blooms through either changes in temperature and or nutrient delivery. And so we're going to be doing that by looking at how storms are influencing tributary nutrient inputs to the coastal zone of Lake Superior. And the second objective is really looking at historical cyanobacterial blooms. So we're going to be reconstructing the occurrence of blooms by using sediment cores from Lake Superior and Siskiwit Lake, and I'll talk a little bit about that later. So we're going to be taking the sort of three-prong sampling approach, with the first prong being tributary sampling. And so we're going to be sampling the Siskiwit, the Brule, and the Nemadji rivers, as long as several smaller creeks within the Duluth Superior Harbor area, which is noted here in panel B in the map. And so at all these different tribs, we're going to be looking at pretty much the suite of water quality analytes. So total phosphorus, soluble reactive phosphorus, particulate carbon, nitrogen, dissolved inorganic nitrogen, silicate, DOC, inorganic carbon, as well as total suspended solids and chlorophyll A. And we're going to be doing this summer in 8 regularly spaced sampling events. So 8 different times, May through September, as well as doing some storm high flow sampling. And so we're going to hope to do about 3 large rain and then sort of the subsequent high discharge events at these rivers, which is going to be triggered by high flow on the Nemadji River. And so this sort of range of base flow conditions throughout the summer supplemented by these high flow, likely high load flow events. And then the second prong is going to be some nearshore sampling, where we're going to be sampling water at 3 different depths, so 5 meter, 10 meter, and 20 meters at 12 transects between the harbor and the Apostle Islands. And again, sort of the whole suite of water quality metrics, same as the tribs, but in addition, we're going to be doing temperature, connectivity, chlorophyll A, and phycocyanin. And we're hoping to do that 3 different times this summer and probably next summer as well. And then the third prong is really this coring analysis, which the coring sites are on the panel in the bottom right in C. And so we're going to be doing cores at Siskiwet Bay, Bark Bay, and Siskiwet Lake, doing dating, percent organic carbon, percent nitrogen, C13 isotopes, and then a bunch of different algal pigments within the core to look for evidence of historical blooms. That's chlorophyll A, phaopigments, and then carotenoid pigments. And we're going to be doing that with 2 1-meter cores in the nearshore bay sites, so Siskiwet Bay and Bark Bay, and then we're going to be doing a long core in Siskiwet Lake, which will span a long time frame looking for evidence of cyanobacterial or algal blooms within Siskiwet Lake. And so I tried to keep it pretty quick to just sort of give a general overview of the 3 types of sampling efforts that we're going to be doing to try to identify those climate and tributary drivers of cyanobacterial blooms in Lake Superior. And that is all I have, and can talk about or answer any questions later on. Thanks.
Yeah, thanks a lot, Rob. Appreciate it. So next we'll move on to Jay Austin with UMD Large Lakes Observatory. He's going to talk about a Bark Point sensor mooring. So Jay, I'll hand it over to you.
And I need to unmute. Can you see presentation? Yes. Okay. This is going to seem a little more serendipitous than the other ones. This was not something that a year ago I was necessarily planning. We're going to have 4 surface buoys out in Lake Superior in 2021. An offshore site here, which is part of an NSF project to study deep convection in the lake. But importantly to this group, 3 sites here in the western arm of Lake Superior. These are surface meteorology buoys. This is one of the 4. They are real-time buoys, so we're getting data over a cell modem while the buoys are deployed. We've had 2 buoys in Lake Superior in the western arm for several years now. This buoy here near McQuaid entrance with a comprehensive set of meteorological measurements, water temperature at 10 different depths. This year we'll have a surface wave sensor on this buoy. And then this year we're going to be adding a YSI sonde with algae, turbidity, conductivity, and pH. And then an offshore site with that standard meteorological package, water temperature, 10 different depths, and then surface waves. It's a smaller buoy, so it doesn't have the capacity to carry more stuff. And then finally, what should be of interest to this group is a surface buoy here just off of Bark Point with, again, that same meteorological package, water temperature throughout the water column, surface waves, and then the same package as this first one, except including dissolved oxygen. And so all of that data will be available in real time. So that's all I got.
Great, excellent, Jay. Thank you. I see some questions coming up in the chat, so we'll get to those when we're done here. Let me quick go to my agenda. Okay, so next on the agenda, we have Henry Quinlan from the U.S. Fish and Wildlife Service going to talk about status and movement of adfluvial salmonids on the Bayfield Peninsula.
Yeah, thanks, Matt. It looks like I've got to wait for someone else to stop using the screen before I can load mine.
Jay, we still have yours.
Yes, yeah, I just bailed out.
Great, thanks.
Okay. So this will be a little different presentation than what we've been listening to up to this point. So my name is Henry Quinlan. I'm a fish biologist with the U.S. Fish and Wildlife Service in Ashland here. And this is a project that 4 different fishery agencies are partnering on related to the movement of fish in nearshore waters and tributaries of the Bayfield Peninsula and throughout Wisconsin, tribs to Lake Superior. So the 4 partners are the Red Cliff Band of Lake Superior Chippewa, Wisconsin DNR, Fish and Wildlife Service, and the Bad River Band of Lake Superior Chippewa. And it relates to our efforts through the Great Lakes Fishery Commission to rehabilitate brook trout in Lake Superior waters. You see the objective, fish community objective for brook trout is to maintain widely distributed self-sustaining populations in as many of the original habitats as possible. And there's an emphasis on lake and stream habitat work. So our objectives for this effort are 5-fold. The first is to document and describe adfluvial movement patterns of brook trout and other salmonids in Wisconsin streams. We're looking to provide baseline brook trout population, stream habitat, and water quality data for some key watersheds throughout the Bayfield Peninsula and over to the Bad River Reservation. We'll evaluate the use of tributaries by stocked salmonids, correlate the movement patterns of these fish with the stream habitat and water quality information we're collecting. Water quality information is temperature and discharge. And then we'll be estimating growth of some of the adfluvial fish, exploitation by angling, as well as coarse scale movement using pit tag recaptures throughout the area. So our approach is going to be to utilize pit tag antenna stations near the mouth of tributaries. So as fish either depart the stream and enter the lake or move from the lake into the streams, we will have pit tag antenna stations set up. And you see a couple of examples here in the top right where there are antennas along the stream bed. And as fish pass over those antenna, as tagged fish pass over those antenna, we record a tag number, the date and time stamp that fish make that movement. And at each antenna station, we will have 2 separate antennas, which will allow us to document the directionality of fish moving in those tributaries. So we'll be pit tagging fish in a number of different steps. We already conduct shoreline electrofishing surveys at various locations off the Red Cliff Reservation, Siskiwit Bay, Bark Bay, and in Chequamegon Bay. So those stations will be surveyed again in 2021, and fish will be pit tagged when encountered. Stream index stations are being added by each of the partner agencies, and those surveys will provide us an opportunity to capture and tag fish. And we will also be tagging hatchery produced fish, so brook trout at Red Cliff and brown trout and splake that are stocked by Wisconsin DNR. And then be recapturing fish through creel surveys. So all creel clerks around the basin will be outfitted with pit tag detectors. Our surveys, we will obviously have pit tag detectors with us, and then the antennas will also serve as recaptures if we see any movement between water bodies. We've got 14 study streams proposed, and those are shown here. Our work is planned for 2021 through 2024. And at the end of that 4-year process or 5-year, we'll be analyzing the data and reporting on that and making some decisions as to whether or not we expand our efforts into other locations in the future. I guess you see one of our locations here is Siskiwit River, which I've been very shocked to see the amount of attention that river is getting. So that'll be interesting for us to see what other information we can connect with the information from the Siskiwit River. And Ian Harding is also on the line from Red Cliff. Ian, if you have anything to add, that would be great. Thank you, everybody.
Great. Thanks a bunch, Henry and Ian. So let's move on to the next talk. We have an update on the Bad River Estuary Mapping Project, which you heard quite a bit about last year. And I believe Eric Dantween from USGS is going to be updating us on that. There we go. We can see your screen, Eric.
Oh, good. I haven't done this before. So this will be a short and sweet presentation here. Faith Fitzpatrick, myself, and Paul Renault are working on a GLRI, USGS GLRI project in the Bad River Estuary to help better understand how extreme events, high water levels, and episodic changes in sedimentation and erosion affect the hydrogeomorphology of and habitat of the Kakagan and Bad River estuaries. Those are Faith's words, with a special interest in lake sturgeon habitat. In this map here, the black circle dots will have stage and velocity meters. They're currently all installed, but the two closest to the lake are not working yet. So I got to find time to get out there and figure out what's going on with them. The other red dots are part of a different project in collaboration with the Bad River Tribe, where we'll be installing stage sensors at all these locations, hopefully soon, as part of a different modeling project. But all these data will be used to develop not a model, but a 3D visualization of the water levels throughout the Bad River and Kakagan estuaries. The tasks for 2021, like I said, monitoring with acoustic velocity and stage sensors. I believe the collect remote sensing data for characterization of springs is temperature to see where groundwater is coming in within the estuary. Paul will be doing long profiles in the Bad River and possibly Kakagan. Let's see here. The output would be to provide a 3D visualization of the water levels relative to the topobathy data. And ultimately, I believe Faith would like to consult with fisheries biologists on implications for the juvenile sturgeon habitat and how the lake levels, the higher lake levels, and the extreme events over the past few years affect that habitat. And that's all I have. Thanks.
Great, thanks, Eric. All right, let's move on to our final talk here, lighting talk for this section. And Xin Wu is going to talk about some nearshore hydrodynamic project updates. Xin, I see, we can see your screen, so I'll hand the mic over to you.
Okay, great. This is the last talk, hopefully meeting your time. So we have two projects we want to update, particularly the first project is related to dangerous current. This project starting from 2019 and continuing working due to the COVID, probably will be extending 1 more years. As you can see, this project really looking at the dangerous current, particularly that characterizing where the location. And there are many people, many folks that are involved with this project, and I cannot list all of those, but these are the main starting team, particularly on the 2 CYAN, funded by the 2 CYAN programs, Minnesota and Wisconsin. And we have 3 locations. One is particular at the Luz and the Wisconsin points along that whole shore on the southwest at the Lake Superior. The second one is a parcel. And the third location is particularly working with Matt on the Chihuahua Bay. And so overall that, as you can see that the project is characterizing and then quantify the recurrent occurrence. Of course, quite a lot of measurement, just so many measures has been deployed, including the wave, into the water level, including the current and some of the drone data. So all of this information, that the unique thing for this project is not only doing this characterization, also working with the National Weather Service that come up with the prediction and working with the community that listed on the whole logos. And I just so happy to, so fortunate to have an opportunity working with so many people that on this project, even during the COVID. So I have to say that if I miss any of those, and not because I forget about it, just so many people, I just want to feed it. So this is the first project, and it will be continuing for this coming summer. We will work with Matt and others, particularly on hydrodynamic observations. And the data will be provided for the people who might be interested in other aspects, like the water quality or algal blooms. The other one is a WISWatch project, also going to happen in the summer. And what you will see from here is that we are currently working with a company that particularly that new spot that we call, and that company that working the new technology that as I can see, if you can see from that, then the spot not only measure the wave, also temperature, wind temperature, as well as now we are going extended to water quality sensor. We are going to the code and doing the compatibilities. Hopefully in the future we'll all incorporate together. And the people are not listening to DNR is that somehow that we also have a conversation with DNR, make sure that whatever we do from this piece, particularly looking at dangerous wave, is not the current, but we like to kind of expand it out, probably related to water quality. If there are interest or ongoing effort, maybe work together on this piece. Of course, there are a lot of people, and I have to say that the whole project is supported by coastal management as well as the CYAN, and started to begin with. And so far, Todd and as well as Nelly and so many people that will probably quickly get together to talk about this project. How do we want to do it? But overall, you can see the picture. There are 5 locations. That's what we want to do. Of course, we were also talking a conversation about possibly deploy particularly at the mouth of the Chihuahua Bay. This one additional. So with that, I'm going to go into this one. So as you can see, this one on the particular on the Chihuahua Bay, I will just give you this sort of opportunity. You can always see this kind of real time. You can click it, and I have this PowerPoint that made you are welcome to share. People just click it, you can get all the information. So this is the date that we have a few days ago. What I was kind of surprised that there is a lake meal. People living in the Chihuahua Bay probably see the lake meal circulation quite a lot. So it's somehow captured it, but you can see the 2 images that eventually will start to stitching together too. So on this one, particular working with the city of Ashland, we also deploy the horizontal ADCP, of course, other sensor. I'll just call your attention about the horizontal ADCP for you to see this. So this is the data we have on water level, current speed. I'm going to go a little bit quick, but just for you to watch that horizontal ADCP, they're changing quite a lot. Particularly, you will see the speed, the direction changing it. You might wonder about why the direction changing it, and what causing the direction changing it. So we see this kind of interesting thing. Of course, we tie with this biodirectional flow with some kind of looking at that one. By every time you see this larger, this kind of bidirectional, then it seems to me there's some kind of storm go over. So we were kind of intrigued about, well, why is this storm go over? And then you can see this kind of thing. Every time we see this, all storm go over. And by the way, the picture you see from the animation here is that these are radar reflectivities anyway. Matt. Okay, so you see this kind of oscillations 2 feet is amazing. So of course we do a little bit, and then we go through a little bit more modeling to figure out what causing it. Of course, this is what convection storm or very narrow band with high pressure that jumping it, as well as the wind speed changing quite dramatically. And I will not like to show this one. I will show it simultaneous for you to look at it. And then as you can see this one, though, we found one very important thing, not even interesting, important thing. Now the storm passed already on Shuagang Bay. And you will see the storm is already long past. Okay, you will not see it. But now, as you can see from later, the Shuagang Bay, that simultaneously you will come with one a little bit later. Now, all of the storm that come up with that effect because of physical mechanism, I'm going to talk about the details. You will see that once that becomes the largest and larger. I'm going to go quick about this one. You will see quite a lot of larger stuff that after the storm. So I'm going to give a one. The large water level fluctuation occur hours after the storm. Most of people probably say, "Fear, okay, fine." But what this kind of a thing that can be very important about meteor tsunami causing it is the way meteorology generates this kind of water level fluctuation. Shuagang responded, water level responded later. And in particular, you will see this kind of fluctuation. That's why the dangerous current. And we see this kind of dangerous current fluctuate back and forth after the storm passed by. Most of the people probably not aware of the danger, and that's what drowning can be possible. Potential drowning can possibly occur. So these are kind of important things we want to do. Of course, then we go to ask ourselves, are we able to do this kind of thing, particular circulation on hydrodynamics? Now, currently, you definitely see this kind of a hydrodynamic. I'm going to show the animation at this point. You will see from this one on the whole lake, but you also can zoom in. You will see that's what the current modeling, particular provided by the girl. That's what the best so far they are doing. So by that way, though, I was just thinking about, well, currently we are working with Google, particularly on the Google survey called Gooser. That particular, I want to show it, and that's very quick moment for you to see that.
So Chin.
Yep.
Chin, it's Matt.
Yep,
You're
okay.
over time here, so
I will go out and that show for you about there's a Gooser. You can see it. But now, so far, you definitely can see this kind of thing for you. If you are any interested on particular on this piece, that you will see the Shuagang Bay, or you can see this piece. But what I want to bring your attention is that we are working on currently on the version about we call core. That project is about, so far you will see that if you have the chance to click it, you will see the surface, you will see the middle, you can see the bottom. Particularly, you will see this kind of core and there's 3 different layers. Working on particular on every locations that particularly on either, if you can see from here, I'm going to switching it, a parcel, I'm going to switching in, and also the what, particular on that the loops. And this all it will be a real time and also focusing in the next time. Thank you.
all right, thank you, Chin. Lots of really great animations there. All right, everyone. So we've reached the point here where we'll take another short break. So I think, thank you very much to everyone who has taken the time to present something to us today. We really appreciate it. It's always amazing to me how much really incredible work is going on in the nearshore areas of Wisconsin in Lake Superior. So we'll take a short break here till 11 o'clock. And, oh, excuse me. Let me back up a second. Why don't we actually do have a couple of minutes for questions. So I didn't mean to skip over that part because I did want to give everyone a chance for questions for our second half of Lightning Talks here. So we actually do have a few minutes for questions before we do a break. And I also want to remind folks that if you haven't filled out that Menti poll that Erin sent out to, I think she has the link in the chat to take the time to do that as well. But real quickly, do we have any specific questions for the Lightning Talk presenters that we just heard from? Feel free to unmute yourself and speak up.
Hey, this is Tom. A question for Eric. He mentioned the remotely sensed data that was collected. I was curious, was that aircraft-based collection of thermal imagery, or what was that? And for,
I'm not sure. I wanna say Faith's plan was to do drone with thermal.
okay, just curious.
I could check with her and I could let you know.
No problem. Thanks.
Yep.
I am looking back through the chat here. I see a question here for Jay Austin. Will the data that you're collecting be going to NBDC in real time?
So the meteorology data and surface waves and water temperature all goes to NDBC. I'm not sure that they have a way to ingest the more biological parameters. Those will be going to GLOSS, who's funding this buoy. This buoy was bought, we underspent in 2020, as you might imagine. And so we're turning some of that into this buoy. And so they'll have the data. And if people want to establish an informal sharing of that data, we'll do it through our website here at UMD.
And the MBDC, is there a link for that you could share with the group?
And NDBC, I can share the link. It's just that.noaa.gov.
Okay.
But again, I'll share the link to our buoy page. That might be a little more helpful if you're interested in more of the biology.
Yeah, that'd be great. Thanks, Jay. Tom Holland, I see you raised your hand.
Hey, I did. So I'm curious about Bark Point, and it seems like there's a change that occurs from the west to the east side of Bark Point. We have the detailed bathymetry that Jay talked about that's collected just to the east there. Does anybody have any thoughts about that? Is there a significant change in the bathymetry or the hydrodynamics once you get on the other side of Bark Point?
I think you may have put your finger on a really important question there, Tom.
Yeah, Tom, this is Brenda. It seems to me that there is a pretty decent shift in the bathymetry right there. There's a big drop off, you know, on the outside of Bark Point there. And it really seems from the remote sensing imagery that, you know, deflection off of that point, you know, changes the whole nature of the South Shore sediment plume and some of the dynamics along there. So I think a lot of really important things happen around that spot.
And the blooms seem to be more prevalent to the east of Bark Point, if I'm not mistaken.
Right. Yep. We have seen blooms before in Bark Bay and to the west of there, but yeah, the most reports have come from to the east of there. And, you know, part of what we've wondered about in the past is it seems, you know, from review of the satellite imagery that often you see dissipation of the sediment plume start to happen after Bark Point. And so maybe there's just enough improved light conditions that those nutrients can really go to work.
Yeah, interesting.
The kind of bigger blooms that we see.
Thanks.
All right, and if you haven't been keeping track, folks, in the chat, there's been some really good back and forth on questions and answers from the different presenters and participants in the meeting. And that's all going to be part of the recording for the meeting as well, so we can revisit that later. Any other quick questions before we take a short break? Okay. Oh, I see one more question here. Chin, if you could post, there's a question about, can your presentation be posted in the chat or elsewhere? Or I think at least the DOCOS links or links where people might be able to access some of the animations that you were showing. Okay. Well, why don't we just do 5 minutes here for a real quick break, come back at let's just say it at 11:05. That'll be 7 minutes. And I think for folks that have joined us that don't want to, the discussion this afternoon will be, or that later when we come back, will be a little bit more focused on CSMI planning efforts. So if you want to be a part of that, feel free. Otherwise, this could be a good time to peel away as well. Make sure you fill out that Menti poll that Aaron put out there. We do plan to actually, when we come back from the break, just Aaron's going to give us an update on the results from that poll, and then we'll jump into the discussion. So thanks everyone. We'll be back at 10:05, and we'll see you then.
Thanks, Matt. See you in a bit, folks.
All right, everyone. Welcome back. Looks like we still have a pretty good crew with us here. So thanks again to all the presenters that we had this morning. Really appreciate it. What a wealth of information and project work that's going to be going on here in 2021. So before we get into the discussion here, I'm going to give Erin a chance to show us what she found from the Menti poll. Go ahead, Erin.
Thanks. Can you see it?
Yes.
Okay, great. So there were just two, the first two questions were kind of, options were provided for you. So the first is what brought you here today? And overwhelmingly, it looks like, simply learning about nearshore monitoring plans for 2021. I shouldn't say simply because there's so much for me to wrap my head around. And I know some of these could have overlapped, but that's good to know. Also looks like just coordination is important to folks, sharing knowledge and data. So I will go to the next one. Whoops. I think I just can use my arrow key here. Okay. So the next was, what's the top benefit the Nearshore group provides for you or your organization? So I understand that many of you have been involved in this group for a few years. So the question's a little bit thinking a bit broader. So improved collaboration was number one, but all of these really look important to me when I see the rankings here. So knowledge or data sharing came in second, coordinated monitoring third. And this was 25 people responded, and this is going to be open for a couple days. So if you kind of just aren't in this headspace right now, but you are later, or you're bored and you want to do a kind of a quiz like thing, please do respond. It's helpful information for us. And then there was an open-ended questions asking if you had any suggestions for the group to help your work in the future. And I was looking over this, and it seems like two things kind of came out of this. The first was just like important to just keep it going, keep it moving. And I think it's a good moment to pause and thank Matt for kind of playing that convening role and keeping this going. It shows to me that it is important, and if it went away, folks would be missing out on some things. The second theme I'm seeing here is related to a bigger umbrella of communication. And there's some really great suggestions here for how to, even just having access to recorded presentations is helpful. I'll be putting the lightning talks and the introductory talk from the CSMI leads at the beginning on the Collaborative's YouTube page, and we'll make sure you get access to that. There's also, looks like, some suggestions for pre and post field season meetings to kind of share out later. And what was the other communication piece? There was a comment here. Oh, a couple of different people, at least 2 or 3, suggested a way to facilitate more ongoing communication, either with a listserv or some kind of setup. And so that's the exact kind of thing I'm here to help with as the collaborative coordinator. So I think the next step for me would be to follow up with you all and with some options, and whether it be a listserv or like a Slack page or some other kind of system, so that you can kind of stay in touch with each other and keep things moving. So thank you for responding. And like I said, it's open for a couple more days. This last question, especially if you have colleagues who might want to fill it out, you can forward them the link. So thank you.
Yeah, thanks, Erin, and thanks everyone really for responding to that. It's really good to hear how this group provides value to you in the work that you're doing, and I'm happy to continue carrying the torch. And I think in particular we spent a lot of time focusing on CSMI this year, but that's not always going to be the case. There's always a lot of really great work going on in the Lake Superior Nearshore in Wisconsin. And I think it's also maybe worth mentioning that as the CSMI work progresses and moves into the results and report out stage, that I think Aaron would be supportive of this as well, that the Lake Superior Collaborative may provide a important place for either sharing information or perhaps in assisting with the reporting out of the CSMI related work in Lake Superior. All right. Well, I had sort of this last little 50 minutes or so that we have on the agenda had just put 3 bullets in there that Anna Baker and some of the other CSMI subgroup leads we'd sort of honed in on as being important discussion topic points for after the meeting or after we got through the lightning talks. But before we get into those, since this is all CSMI focused, I do want to provide an opportunity for others that either presented non-CSMI related work or have other ideas about would there be a need for further coordination or discussion around any of these sort of non-CSMI related focal areas. And if anyone has anything they'd like to bring up related to that, I think now would be a good time to do that.
This is Kate Reinl. I just want to add that, even though, we've been calling these all like CSMI subgroups and things like that, you might have seen like from Ellen's slide, we've been welcoming both CSMI and non-CSMI work. And both of those types of projects are being represented, in the planning maps. So if you are interested in, collaborating with the CSMI projects, even if you're not funded by CSMI, we definitely want to include as much of this work as possible because it's all important to, the nearshore work in Lake Superior.
Yeah, that's a great point, Kate. And I think as we saw today, there's a lot of related work related to the CSMI work that's not funded directly through CSMI going on here. And I think the fact that a lot of that work is already being collaborated with or coordinated with the CSMI efforts, I think, really shows how well a lot of the folks in this region tend to collaborate and work together anyway.
I was going to comment, Matt, too, about communication and outreach. Kate and I have had a few conversations with Val Brady of Minnesota Grant, who will be coordinating the reporting on the CSMI. We'll also be meeting with some EPA PR people going forward about social media and how we can just spread the word about the CSMI efforts generally to the public. So if people are interested in that, let Kate and I know.
Yeah, I think we would be, Tom. We're planning to do a little bit of work around that or related to that with some of the project work we have going on this summer. So maybe we can follow up offline here.
Yeah, hey, sounds good.
Okay, well, why don't we move into the specific sort of, I guess, CSMI related bullets here for our group discussion. There's sort of the 3 main focal areas that we thought of before this meeting were related to, are there any further needs for sample planning, either between groups that are doing things that haven't been coordinated before, or if there are maybe gaps in locations or gaps in the kind of work that's going on. That's kind of the idea with the first bullet with the group discussion. The second one that we'll revisit after that would be the interlab comparison. I think Kate has a couple of things to mention related to that. And then the third was talking about, well, what happens if we actually get an algal bloom this summer? And so we're putting a lot of eggs in the basket here, that all these resources going towards trying to understand algal blooms in Lake Superior and what's driving them. And certainly there's no guarantee that we'll have a bloom. But if there is, that we want to make sure we don't miss it, and have in the past couple of years, there's been a sort of a phone tree and a response plan for how different groups that are in a position to sample the bloom plan to mobilize if that indeed happens. So I think we can revisit that as well. So those are the three main areas that sort of the CSMI folks felt were important to address in this discussion. Any other lingering topic areas that people want to think we should be discussing here at the end?
Hey, Matt, if I could jump in again, Gerald Keith, I think, is on the call, and I wonder if he could real briefly say how he would use the Secchi depth information for calibrating his algorithm. It seems we have pretty good, a few comments from people in the chat that they're happy they can easily collect that information. If Daryl, if you could just quickly express how you'd use that, I appreciate it. I know he said he had to step out, so he might be here in the call, but attending to something else at the time. May have missed that.
Yeah, well, if not, I think that's in terms of sample planning, it fits right in with that first bullet in our discussion. That was definitely something that the Secchi depth, I think both Bob Sterner and I, as we're doing our nearshore sampling, that's something that we can easily incorporate.
Yeah, and I comment about Bark Point and that she has historical Secchi depth info. It'd be interesting even just to compare the east and west side of Bark Point in terms of Secchi depth info.
Mm-hmm. And we have quite a bit of that information over in the Schwamigan Bay side of the peninsula, going back to 2014. And then I should also mention, Tom, that Daniela Gerlin, who was formerly with the Wisconsin DNR, had been working with NASA on a project to identify, basically using remote sensing to predict algal blooms. And we had shared some of our Secchi depth and water temperature and some other information from Schwamigan Bay. And I'm curious if Daniela and that particular research has been tied in at all with what you're doing, Tom?
Not yet, but we're interested for sure.
Okay, go ahead, Abir.
I was just going to say, you'll notice Kate has a point in the chat which sort of relates to that, and I think it's worth talking about a little bit. It's suggesting that we can coordinate our sampling efforts with satellite flyovers and stuff. So with the remote sensing, I think it's a good idea, especially if we're trying to validate methods or try to assess if we can predict in advance based on what we're seeing at that scale.
Yeah, great point. Well, it seems like in particular, the nearshore efforts, we have some sort of rough idea of sample dates between, I'm talking about at least the Northland College UMD effort and the Wisconsin DNR nearshore effort, sampling dates. But I think, yeah, Tom, having that, if you have that satellite flyover table, I haven't seen that yet, so it'd be good to have that as we plan out the specific dates that we'd like to try to do our nearshore sampling.
Yeah, we'll get that up to the hub site really soon. Happy to
Yeah, on that, it'll be great to see those dates. We have to also coordinate with people and boats and a lot of other things. So there's a lot of moving parts there, but knowing when the satellites will be overhead could be pretty helpful.
Is there a specific satellite that we'd be prioritizing to try to catch? I noticed there was quite a few different ones, Tom, on the list that you sent.
Well, in terms, it's hard to say. Weather is such a big factor in clouds, in that the Landsat 7 and 8 will be really nice in terms of the resolution, as is the Sentinel. But I find, what I've looked through so far, the Sentinel 3 sometimes has the more striking images where you really see the sediment along the near shore. I think Daryl Keith is focusing on Landsat 7, 8, and the Sentinel 2.
Tim Asplin just put in the chat, the Sentinel are designed to capture water quality.
Tom, is it all that stuff that you sent in the email to me and Ellen, the satellite dates 2021 file?
Yep.
Okay.
Shared that widely yet. I just wanted to double check it, but I'm pretty good with it now. But we'll get it out. We could send
I
it.
just wanted to note, Daniela has moved on. She's moved back to Germany. She's no longer working with the DNR. She's still got a volunteer status, but we really don't have the in-house capacity as much as we did when she and Steve Greb and others were here for tracking this information. But I know Gina Liberty is kind of connected with NEMA at NASA, and so that would be good to see this work continue. I think there's a lot of really useful applications here, but I don't see this DNR being kind of in a coordinated or leading role on this, at least for the near term.
One of the things that we didn't mention is Noah Sigler will be bringing a wave glider to Lake Superior. They're going to keep it to the east side of the Keweenaw Peninsula, and that's going to be informing a productivity model that Michigan Tech is developing with Andrea Vanderwood of NOAA using remotely sensed data.
And it looks like Gina Liberty shared Nima Palevin at NASA contact information there, who was working with Daniela that Tim just mentioned. Looks like he would be interested in data collaboration, especially for sampling coinciding with the satellite dates. So Tom, that might be a good connection to make.
Yeah, sounds good. Thanks, Gina.
It would be good to kind of get, I guess, a list of names of who would like to adjust their nearshore sampling to the flyover time. So if you want to throw your name in the chat so that we can collect those names, that would be helpful for Tom and I.
Great, thanks, Kate. I know, and Bob just mentioned it, so the work that we'll be doing with Bob, we're interested in the coordination with the satellite overflights for sure.
Have you guys nailed down your BRICO sampling locations?
I think they're pretty close. Ellen gave her talk. There were some locations that were on the map she gave, and Bob has a Google Scratchpad that has all those locations on it. So I think we're pretty close. Jay just mentioned in the chat he'd encourage adding a sampling location at the surface buoy there off Bark Point to help in ground truthing its sensors. That's one thing that I think Bob and I are talking about, whether or not to add a station there to fulfill that purpose. But I think we're pretty close. It's just the final tweaking based on resources we have versus number of samples locations we can have and timing. So I think we're pretty close.
Yeah, it'd be great whenever you guys kind of finalize those. I'll add them in with all the other sites and get them mapped out so we can get that coverage shown as well.
Yeah, you're right. That is one of the missing things from that ArcGIS map at the moment.
I guess while we're also talking about planning, if there's anybody who's using that map for planning, especially from the Tributary group, I've gotten a lot of input. If there's something that you need on that map to help you plan, like a different layer, so like one of the requests I had was for higher resolution NHD data. If you need something like that, including like geology, soil erosion layers, things like that, I can add those things to the map. So if you need anything like that, just let me know.
I was just going to follow up on Kate's comment too, that the hub site, if folks have something they're interested in sharing, we can easily plug in additional pages into that site. There's a lot of different interlinks and mashups and things that we can do. Matt, I emailed you that spreadsheet with the satellite overflights. If you could share that with this full list, that would be great. I think you blind see the announcement, so I didn't have that.
Oh, right. Yeah, I can do that when we send out some meeting summary following the meeting.
Thanks.
Okay. I did have one item that didn't come up, at least I didn't hear it, as part of the nearshore CSMI planning, was glider work through EPA. And Tom, is that's still planned as far as you know, as part of the CSMI work?
Yep, we're planning a spring flight or spring mission for probably 2 weeks, and then a late summer. For sure with our G2 glider, and then hopefully also with our new G3 glider that we have not yet had in the water.
Paul McKinney provided glider transects on that map. I can share it really quick. Proposed glider transects. These purple dots with the line in between them, those are the proposed glider transects. I know that there's been some talk about, you know, even lining up some of this stuff, like seeing if we could line up the glider transects with some of these, you know, discrete water sampling transects. And I know Paul was open to, you know, integrating it in that way.
And we've flown this area quite often up to Bark Point. And that's available on the glass glider DAC. If anyone's interested in that, let me know. We can get you in touch with that data. Going back to, I think, 2014. Previously, we turned around at Bark Point, but I think learning after what we've talked about today, we'll want to continue on past Bark Point a fair bit with the glider.
Could you refresh my memory? The new glider has multiple fluorescence probes, is that correct?
Yeah, it has also, in addition to the phycocyanin sensor, and then a list sensor for particle size and concentration.
Cool, thanks.
And Tom, the work with Bob, you know, our furthest offshore site that we're hoping to get to is somewhat designed to be at least, I think, 20 meters of depth. So to try to connect, you know, into depths where the glider would be, transects would be going. So trying to connect some of that near shore to offshore work. So it'd be good if at least one of our or two of our sampling events later in the summer could coincide with when that glider is out doing its late summer work.
That'd be great. As soon as we have a date, yeah, we'll let you know. Which makes me think that we haven't put an exact, we have sampling dates on these points in the map, but we don't have like a cross-reference table yet, calendar or a NAT chart yet of sampling. Maybe perhaps that would be useful, so we can see when people overlap and so forth visually pretty quickly.
Yeah, we haven't gotten any kind of like firm dates. A lot of these are kind of, you know, what it has, like, for example, in this glider one is kind of like a rough idea, 3 weeks in May and August, not any kind of firm dates. So if there's some updates that we can do to this to firm it up and, you know, really nail down those dates, that would be great.
That would be good. This is Anna. I like your idea, Tom, too, of having some kind of table where you can see just overlapping. Here's the window of time when we plan to potentially be out, just so that we can get a sense of how timing aligns.
It'll probably be that satellite data, right? That'll drive a lot of that. So once we get that information, we should be able to put a calendar together.
Yeah, I think so, Ellen. I think that's a good idea. Would it be helpful for us to put together like a Google Doc where we list all of those satellite dates, and then we can kind of have a column for each group to kind of just throw in their own X's of like where they would also sample?
That could help.
I think that'd be great.
Yeah, I think that'd be helpful.
Mhm.
The other thing I'm curious about too is just previous years and previous storms and the dates of those, and if there's, you know, if we have, I don't know, any kind of idea of when we, you know, which months we might expect the biggest storms.
Hey, Tom, if you look at our L&L paper, we have some hydrology data like that going back to 2011, I think.
Yeah, okay.
So it would be through 2018. But it has flow and precipitation data that we gathered for that paper.
Excellent.
Kate, you could leave that map up there if you don't mind.
Oh yeah, I'll pull it back up in just one
It-
second. I was checking another map because I have the so I have live I almost said stream gauge data on another map actually. And I think you might be able to look at the older data as well. So I was just checking that really quick, but yeah, I can pull up that map again, just 1 second.
Okay.
Let me see.
Is anyone, you know, with regards to a large runoff event, you know, just thinking about all the rain we got just last night in our region, I don't think it was large storm event kind of material, but you know, those things can happen at any time. And is anyone planning on adjusting sampling activities around? Let's say we get a large storm event at the end of May or middle of May. You know, right now our thought is sort of beginning sampling around the mid-June time frame, but if we get a big event earlier than that, are people in a position to mobilize sooner than that, or just kind of stick with a mid-June sort of time frame?
Matt, I don't know if you're referring to nearshore sampling specifically, but the tributary sampling is starting now. Eric got our gauges installed yesterday. Hooray. So we're hoping to, our work is very storm driven. And so we're hoping to be capturing flow and water chemistry across any storm peaks that happen from basically here to October, in addition to baseload conditions. One of our challenges will be kind of how to distribute our sampling. We want to capture the big events, but we're also really interested in how those events drive blooms. And so having some ability to predict what kind of storm magnitude and what kind of temperature and ambient conditions might lead to a bloom, so that we can potentially try and sample in between storm and bloom, or at least capture the storm itself, and then when the bloom occurs, be sure we get back out there again to get another sample. That's kind of one of the coordination challenges that we face. We will be somewhat limited in the number of samples that we can get, but we are mobilizing now. So hopefully that's helpful on the tributary side, at least.
Mm-hmm.
I don't know if Eric has anything to add to that end, if he's still on.
Matt, the Nemadji work that we're doing in cooperation with the NER, the NER will be putting their ISCO out at the USGS gauging site in the Nemadji. And the plan we have with them is to anticipate storms based on weather forecasts and program the ISCO to sample those events. So it'll be somewhat of a kind of a responsive program relative to what we think the weather is going to do in the coming day or two. So that's part of what you were talking about. I don't think we have an agreement with them yet exactly when that work would start. So whether it, is going to happen in May or not, I don't know for sure, but it will, that NER cooperation on the Nemadji will definitely be storm driven sampling.
Mhm.
And as much as Wisconsin DNR would like to be flexible, we're not going to be able to probably mobilize too early because we're not able to hire our LTE position out until mid-May or so. So we probably won't have the personnel to be able to get out there early.
Yeah, I think this is Matt again. I think we'll be somewhat in a similar position, Ellen. I mean, once we get into June, I think we'll be okay. But before that, we'll have some capacity limitations, as well as trying to get everything in order in terms of materials and I think a strategy with Sandy and Bob over at UMD. I think we're meeting in a couple weeks here to start working on that. But we'll be a little flexible, but not too much before June. But I guess, if we do end up with let's say a large runoff event in mid-May, that kind of, depending on how big it is, those kind of things tend to persist for quite a while. And there's also that, lag time that has been identified, between events and blooms. So I think we'll do the best we can, and maybe the best thing we can do is be in coordination with each other as the summer goes on, and that maybe gets a little bit into the third bullet on the list regarding algal bloom response. But it's maybe more than just, do we have a response team, but it's sort of like an event response or sampling response team. Maybe a more coordinated approach that we can use to mobilize people when there's something we want to get collaborative or collective data sets from different areas at the same time. Looks like Kate stepped out for a couple minutes, but that was a little bit of a hint to Kate. I know we had a Google Docs or a spreadsheet that she had organized the past couple years with a phone tree and sort of a plan for if a bloom was spotted, who was going to talk to who, and how we were going to work to get boats on the water to try to get samples. So I think the idea was to try to reignite that Google spreadsheet and make sure we got all the people on there that would want to be notified to mobilize sampling activities.
And that makes a lot of sense.
Yeah, Matt, I think that's right. And I don't know when our next algal bloom subgroup call is. I was just going to look at my calendar. Is that one the first Monday?
I think it would probably be in a couple of weeks.
That's what I'm thinking. Seems like it's coming around.
Yeah, I think that's a good topic for that next algal subgroup call is let's figure out our response. But it's not just bloom response. I think it's also the storm event sampling response.
Hey, Matt, if you want to spend just a few more minutes on this topic, I have pulled up on my computer that hydrology figure that I indicated to Tom, and it shows when events, rainfall and flow events have happened. So I could share my screen. It would take, 2-3 minutes of discussion probably to get through it.
Yeah, go ahead, Bob.
Okay. So there, I believe you have it.
Yes.
So this is, again, this is published, so anybody can get to this. This is precipitation where we looked at 3 weather stations in the South Shore between the Twin Ports and the Apostles, and we averaged those. So it's kind of a South Shore wide indication of rainfall and what is it again? 3-day moving averages. And then what we did is we colored June, July, and August in red. The blue triangles are the 2 big 2012 and 2018 events. So this is rainfall. And then the flow data from USGS gauging stations in the Nemadji and the Brule and Whittlesey Creek. Nemadji is way, way bigger, of course, than the Brule, which is bigger than Whittlesey. So, I mean, you can see some years snow melt is really the big event. That's what I think is going on here. But like in the 2018 bloom was, you know, a really significant flow event, which, you know, really spikes up above the rest of the data. So, and then this table here is the top 5 events for rainfall and flow across all those years. So June 18 was June in 2012 and June of 2018 were those 2 kind of singularly large events.
And then July 2016 was, now that event didn't hit the Nemadji, the Brule, or the Whittlesey quite as much. The Bad River received the brunt of that event. That was a sort of mid-July.
That's right. And so it doesn't stand out in this compilation because, as you said, it was not really historically large in this particular segment of a watershed that this graph is referring to.
Bob, this is Anna. This is really helpful. Thank you. Can you remind us when in time the blooms occurred relative to these storm peaks?
Yeah, they were so mid-July of 2012 and 2018 was later, August 9, 2018. So it was, I'm going from memory, Anna, we'd have to look at the paper again, but it was something like, I don't know, a 3 to 4 week lag in one case and more like a 5 to 6 week or longer lag in the other.
Yeah, that's right, Bob.
Okay.
Thank you.
Yep. I'll stop sharing.
Yeah, thanks, Bob. Yeah, I also noticed, I mean, we certainly have the spring snowmelt peak, which has already occurred for this particular year. And I'm not sure it was a particularly epic spring runoff this year. And then I noticed too, and we noticed this in some of our data, that the fall October timeframe has been a big time for events too, going into the winter. But it seems like from what you're showing us, you know, we should definitely be ready to go by early June. And I think that's kind of where people are at now. Well, I'll send a note to Amy Thomas about adding sort of this bloom response, sampling response question, and trying to get into the weeds on that a little bit more at the next algal subgroup call, which Tom posted in the chat is April 26th. Those are, I believe, at 1 o'clock Central Time. So if anyone needs information about how to join that call, just let me know, and I can get you in touch with Amy. Kate just posted a link to satellite flyover, sample timing, Google Sheet for planning. Thanks, Kate. All right, well, anything further on the, I guess we've sort of hit on the sample planning and the algal bloom response plan. I feel like we've got a good next step regarding that stuff. This is kind of what happened last year too, is we had this Nearshore meeting, and then a number of the discussion items moved to the algal subgroup afterwards.
So I'll just remind everybody, the really big event in 2018, we got a phone call from Brenda one evening. Sandy, I seem to recall we were having a lab party at my house or something like that, and Brenda called and said there's a bloom, and we were able to mobilize the next day, and really like within 3 days it was all over. And so when we talk about mobilizing for blooms, we're talking about like getting out there within a day. These blooms don't last long in this environment. And even right now, I would say we can't point to a single data set of any kind which clearly shows bloom life history or progression. Sampling's been opportunistic. So I mean, all of this work that's being planned is really, really awesome. And if there is a bloom, I think we're going to be in much better shape this year than we have in the past. But do keep in mind, these events are quite ephemeral.
It's a great point, Bob. And I think all the more reason that we need to really nail down how we intend to communicate with each other, especially those that can mobilize quickly to get boats on the water.
Yep. And a good idea to have lots of redundancy, like anyone who's able to be on the list, so that we can go down the list, you know, if something happens, and really get somebody to be able to be on the water. Because it seems like whenever these happen, it's like vacation season, people are gone, it's the weekend. There's always lots of complications in trying to get a response going.
Great. Well, I want to make sure that Kate has a moment to just refer to the interlab comparison. She had a note from Joel Hoffman related to that. Kate, are you still on?
Yep, I'm still here. Yeah, so we've talked a lot, you know, I'm sure a lot of you have been part of these conversations, that we need to make sure that our lab analyses are, you know, producing comparable results. So we have all these data sets so that they can actually really be integrated and compatible. So we've talked a lot about doing these, you know, exchanging of samples. The formalized name for this project is the Lake Superior Interlab Calibration Effort, or Lake Superior ICE. And so Joel Hoffman at EPA and Chris Philstrup at NRI are leading the charge on that, and Kansas Keaton is also assisting with the sensor component. So basically, they're looking for people who are interested and willing to participate to please reach out to Joel and Chris. We can put their emails in the chat, maybe at Kansas Keaton's as well. And that, you know, it's not, you know, obviously a mandatory thing, but, you know, for this data to actually be able to be used together, we do need to, you know, really put an effort into doing this. So this would be a fairly formalized effort, not just this kind of like casual, you know, transfer of samples. It would be something that's well coordinated. So I'll add their emails to the chat. That'll be for both wet chem, specifically nutrients and pigments, as well as sensors. And also that, or maybe I think I got all of it actually there. But if anybody has any comments, you know, right now I can relay that or through the chat or whatever to Joel and Chris.
Bob and Sandy, is that something you're planning to participate in with sampling we're doing?
forgot to unmute. You're asking about the interlab comparison?
Correct, yep.
Oh, for sure. We'll participate. We're not in a position to drive the bus, but we're definitely on it. We in fact collected samples last year. There was a kind of a nascent effort to try to do this last year, and I don't think it ultimately resulted in a interlab comparison. That could be wrong, but we're definitely able to collect samples and trade samples and stuff. This year, I think it's really important to do that.
Yeah, the effort last year resulted in some comparison samples between the Burke Center lab and your lab, but I don't think this with the state lab. And so we're not doing any sample analysis in our lab this summer at the Burke lab. So I think it's really the comparison with the state lab and other labs that is going to be most important, at least for this year. Okay. So I'm coming up with just a list here of things to sort of send out and follow up to this meeting. And maybe you all can help me make sure I'm not missing anything. So I have, I'll just include a note about the interlab comparison study and making sure to reach out to Joel Hoffman or Chris Philstrup with respect to anyone who's interested in participating in that interlab comparison effort, which includes wet chemistry and SOND data. I'll put a note in there about the upcoming April 26 algal subgroup and the need for sort of phone tree type coordination of sampling efforts that will hopefully occur there. Bob Sterner included a kind of a summary of the various projects they've got going on that I can send along. He asked me to do that, as well as the file with the satellite over flights of the satellites that we can send out for planning purposes, and then the summary notes from the meeting as well. Anything I'm missing for that folks would like me to include as I send out a follow-up communication to the group?
Did you say you're going to save the chat too? That'd be helpful.
Chat is being saved, yes.
All right.
I'm gonna save it and kind of synthesize it a little bit, and we can make sure it gets back to everybody.
Thanks, Aaron.
Yeah, thanks, Aaron. And also to Val Damstra. Val's been working on the same kind of thing, working on notes and behind the scenes kind of stuff. Okay. Well,
I'll comment briefly. You know, I was involved with the Lake Ontario CSMI effort, and after the field season, we followed with a fall data meeting for people to describe and present the data that they had collected in some of their initial analysis. And at that point, the group, the Lake Ontario CSMI group, was able to pretty quickly outline 9 to a dozen different papers that people were interested in bringing from that data. That led to a special issue in IAGLR. So I'm very hopeful that there's going to be a lot of interesting data collected and opportunities for manuscripts and papers, and suggest that we start thinking now about what those papers might be and planning for a data meeting come sometime in the fall or next winter.
yeah, thanks a bunch, Tom. Is there any sort of planning underway for that, or a group that's thinking about that so far that you're aware of?
Not yet. The Lake Ontario came about pretty organically, and that happened.
From our side, we have done such a meeting just for our group and with Brenda at NPS pretty much every year. We're typically ready to do that late winter. There's enough analytical work in the lab that takes a while. Then we have to kind of comb through the data a little bit. So I definitely be game to participate in that, February-ish, something like that.
Since I'm leading the data integration piece, I can take ownership of that and plan a data meeting. We'll find a time that works for everybody after they've got their data in and analyzed and all of that good stuff. Because we're going to need to have some sort of meeting like that anyway, just for the data reporting piece.
Okay. And that, I think there's at least potential either through this group or the Lake Superior Collaborative in general. I can't speak for them, but we do have, a couple of forums already, the algal subgroup and through the LAMP that are existing that I think would be willing to assist in that as well. February of next year is, I think, a real good time to look at doing something like that.
I think we can also anticipate a following State of Lake Superior meeting too, supported by Agler, as they've done with the other CSMIs.
Now that would most likely be a couple years. That'd be like 2023.
Yeah, I'm not sure how. Yeah, maybe 2 years out. Yeah.
I think that the meeting that we're talking about here would be something sooner than that, to sort of
absolutely.
post field season, looking at how we want to integrate or look at coordination on publications. Yeah, next State of Lake Superior is 2023, says Val. Okay, well, I look forward to that. That'll be after a very exciting and event-filled summer where a lot of data are generated by a lot of different folks. It's really impressive to see the amount of work that's going on, and hopefully we don't trip over each other too much while we're in the field and we're as coordinated as possible with these sampling efforts. Any further comments that folks would like to make before we wrap it up here today? Make sure to high five each other on the boats as we pass. All right. I really appreciate everybody joining in today. We've had, once again, a lot of really great discussion and a lot of great participation from folks. So thank you so much for being a part of it. Thank you so much to Aaron and the Lake Superior Collaborative for hosting the meeting today. I think it's been a really great first go with the Nearshore group being part of the collaborative, and I think it went really well. And look for some follow-up message from me here in the next few days, just summarizing the meeting and getting out some of that information I talked about. And certainly look forward to talking to a lot of you again on the next algal subgroup call on April 26th.
Thanks so much, Matt and everybody.
Thanks, Matt. Matt and Aaron, yeah, thank you.
Thanks, everyone.
You're welcome. Thanks, everyone.
Yeah.
Take care. Thanks.